Hey guys, welcome back. This is going to be our third and I think final video for Chapter 9, DNA to Protein. I left you guys off last time with this slide going transcription is DNA to RNA. Now we're going to talk translation, RNA to the protein. So, the first thing you need to know is that all of our different types of RNA work together. Transfer RNAs, you saw it in the lab, deliver amino acids to our ribosomes. And transfer RNAs contain this very important thing called an anticodon. Anticodon is complementary to the mRNA codon. So the mRNA codon is UCG. Then we would have to go find the codon, the anticodon that matches those. So U bonds to A, so you have to find A. G bonds to C, so it has to be C. And C bonds to G. So we'd have to go out into the tRNA world and find ACG, right? And ACG would be attached to a specific amino acid. And so that's tRNA. That's how they work. They have that anticodon. They also contained a binding site for the amino acid. That was that little stick that you had to poke into like a ball in the lab there. And that's coded for by the codon. The rRNA, different type of ribosomal RNA, and proteins, of course, if you don't know, guess protein, make up ribosomes. And you guys know what ribosomes do already. They make proteins, that's all they do. So they link amino acids into polypeptide chains, which is a wordy way of saying making proteins. And two ribosomal subunits in proteins make up each ribosome. Sometimes biologists do it right, and here I think they've certainly done that. They have named the bigger piece of a ribosome the large subunit, and they've named the smaller piece of the ribosome the small subunit. So that's kind of nice. We just call the big one big and the small one small, and we don't overcomplicate things. So there's a ribosome. Oh, pardon me. So here's what one example of a tRNA might look like. You have your anticodon ACC. So here they are, ACC. That's what's actually going to match with that mRNA strand. And then here you have this long structure of ribosomal RNA. And then at the end, you have what I think is a tryptophan. And so there's a tryptophan right here. That's your amino acid. And that's what's going to be connected to the protein. So translation converts genetic information from our mRNA into a new polypeptide, so our new protein. The amino acid order is determined by the codon order, right? So hopefully you're getting this kind of trend. The sequence of the DNA determines the sequence of the RNA, which determines the codons, and the codons determine the amino acids. And so another thing that's really important you need to know about translation is where it occurs. This stuff occurs in the cytoplasm. Okay? Transcription was in the nucleus. Translation is in the cytoplasm. And it occurs in 3 stages. Initiation, which is the tRNA actually attaching to a ribosome in the correct location. That's initiation. And then elongation. As the ribosome moves along, it's making a longer protein strand. And then termination, where that amino acid strand is broken, and it is hopefully a complete protein. So there's your 3 stages. And so here it is, transcription, DNA. We made the, what they call it, the transcription bubble. I love that name, that's funny. Transcription, and then the messenger RNA comes out here, and then the initiation of the ribosome subunits onto the mRNA. They cruise along, they make an amino acid strand, which is our protein right here, and then when they're done, they'll let go, and the protein will leave, and they'll disassemble and run back here and grab a different strand, maybe one of these guys that's being made. So that's how that works. So initiation. As we said, there was initiation, elongation, and termination. We're going to go through all of them. The initiation is the initiation complex forms. So the small subunit of the ribosome binds to messenger RNA first. The anticodon of a tRNA base pairs start with the AUG of mRNA. So, quick question. If the codon for mRNA is AUG, what's the anticodon for the tRNA? Alright, well, hopefully you looked at it and said UAC would be what's on the tRNA. That's the anticodon. And so we always start with methionine, and AUG is the start codon for methionine. And so once we've got the small ribosome unit, once we've got the anticodon, then we add the large ribosome subunit, we've added methionine, and we are ready to make our trip down this messenger RNA strand. So not too complicated. Elongation, next step. The ribosome assembles the polypeptide chain as it moves along, just like you did in the game. The ribosome moves down the messenger RNA, assembles a new polypeptide every step, and the initiator tRNA carries methionine, like I had already mentioned for you. It's always the first. The ribosome joins each amino acid to the polypeptide chain with a peptide bond. And you guys learned, I think in chapter 3, that proteins were held together with peptide bonds. So now you know. That's what holds together proteins. And the ribosome is what makes that bond. So termination. When the ribosome encounters a stop codon, it's a codon that doesn't code for anything, the polypeptide synthesis ends. So there are no more amino acids, which means we're done. The release factors bind to the ribosome. The enzymes detach from the mRNA, which is the small and the large ribosome subunit, and the polypeptide chain breaks away from the ribosome, and it's a done deal. So that's how we let go of messenger RNA. If you don't know, guess protein, and here you'd be right again because it's enzymes that are actually pulling the ribosome off of the RNA and pulling the protein off of the ribosome. So there's somebody for every little job. And if you don't know, guess protein, because they do almost everything. Alright, so here's your picture of the whole process. Ribosome subunits and the initial tRNA converge on the messenger RNA, that's the brown guy right here. A second tRNA binds to the second codon. We form a peptide bond right here between methionine and valine. And then as the ribosome starts to move along, our next tRNA comes in, looks like leucine. Here you can see UUA, AAU fits in here. Leucine bonds to valine. On and on and on she goes. Where it stops, well, let's stop. I guess somebody knows, stop codon knows, but there you have it. So we just keep going down the line until we hit a stop codon. And there it is all together. At some point we hit a stop codon, UAA, and we go up, time to fall apart. Let go, let go, let go, protein. So, let's get to the fun stuff. Mutations. Mutated genes and their protein products. If the nucleotide sequence of a gene changes, it may result in an altered gene product with harmful effects. So this, we've been looking a lot at RNA, but just so you know where we're going now, nucleotide sequence of a gene, we are talking about the DNA, not the RNA. So we're going way back to the beginning of this chapter. If you change a base in the DNA, you can change the protein. So mutations can be small, really small scale changes in the nucleotide sequence of the cell's DNA, and basically they're going to alter our genetic code. Now, they're pretty uncommon events in normal cells, unless you're being hit with X-rays or UV rays from the sun. Most of your cells are going to be really good at replicating DNA exactly the way the template was. And even when mistakes happen, we have a little proofreader enzymes from last chapter that come along and make sure everything's cool. And so, mutations, they're pretty rare. Unless you're like, I don't know, smoking a cigarette in a tanning bed. Okay, then they're not that rare. Then you're asking for mutations. But most of the time, your cells are pretty good at taking care of this. A mutation that changes UCU to UCC is silent. So you remember when I told you that there were, what, 64 codons for 20 amino acids? And I said that that created redundancy? This is that redundancy point being applied. We changed a base, UCU to UCC. That could change everything. That could be fatal. But it's not, because it's redundant. UCU and UCC code for the same amino acid. So even though we changed something, our protein is the same. And so we kind of wipe the sweat off our foreheads and say, whew, we're fine, okay? It has no effect on the gene. So we're in good shape there. Other mutations can have a severe consequence. If they change that amino acid, our protein's not going to be the same shape. Shape determines function, and this could be anything from not a big deal to fatal. It just depends. It can result in a premature stop codon. That would be a bummer. If you change a base around, you might end up getting a stop codon by accident. And even though this protein is supposed to be very, very lengthy, when the ribosome hits that stop codon, it lets go. And so you end up getting a protein that's half the length it should be, or 3/4 the length it should be. And it's not going to be able to do its job if it's not the right size, because it won't fold into the right shape, and shape determines function. Cool? So, a couple examples of this. One is the base pair substitution. Okay, so a base pair substitution is if we had, I don't know, AAG, and we pull out the second A and replace it with a C. Now we have ACG. Okay, totally change the base. This can result in the stop codon, a premature stop codon, which would be pretty bad, or a different amino acid in the protein, which would be pretty bad. One example of this is sickle cell anemia. Okay, so you can check that out in your book, but basically the red blood cells get this long sickle shape, and they get stuck in capillaries, and that's not good. So the other way to do it is deletion or insertion. And so here you can imagine you had AAG again from our last example, and we just pull the G out. Just by accident, we just lose the G. Okay, now when you have AA nothing, it changes your reading frame, which means you read everything in a block of 3. So if I remove one nucleotide, if I remove one base, nitrogenous base, right, then, or I add one nitrogenous base, I'm going to either shift everything you read one space to the left, or I'm going to shift everything you read one space to the right. And this is going to ruin everything. It's terrible. Your book has this cool example. If you go to chapter 9 in your book and flip through kind of near the page of that gross looking cat, there is this really fun sentence that says, "The fat cat ate the sad rat," and it's all 3 letters, right? Every word is 3 letters. So every word is like a codon. And then they do an insertion. So they change the reading frame. And so it's T, and then a space, and then 3 letters, and then a space for the rest of the sentence. And by changing the reading frame 1 letter, the sentence is garbled nonsense. It's not even words anymore. And it's exactly what happens if you shift your reading frame when you're trying to read DNA. So our example there is thalassemia, which is a type of anemia that we'll talk about just in a slide or two. And so here it is. Our examples of mutations, we look at hemoglobin in a human. Hemoglobin binds to oxygen in our red blood cells. And this protein is 4 polypeptides. We have alpha globins in blue, pretty complex proteins, beta globins in green, and each globin has a pocket that cradles this little red group here called a heme. And the heme contains iron, and iron bonds to oxygen in our blood, which if you think about it is kind of why our blood is red in the first place, because iron with oxygen is red, so that's kind of cool. Think rust, think blood, it's weird. So the next part of our DNA, this blue part, and then we have our messenger RNA in brown, and what they end up making for their amino acid sequence, the CCU, the GAG, each codon, GAG again, AAG, and so that's the kind of the code here. So a base pair substitution, here you see we had CTC, we now have CAC. It's not redundant. It happens to make something new. So when we go to pair it up, we get GUG, different than before. We now code for valine. This is a different amino acid. And so valine replaces glutamic acid that we should normally make. And hemoglobin with this form is called HbS, or sickle hemoglobin. So that one little change in that one little base made a cell that looks nothing like a normal red blood cell. It's a huge change. So let's look at D, this example here. A base pair deletion. We had GGAC, GGAC, TCC, blank CC. We have deleted that T by accident. We lost him somewhere. We forgot to copy him somewhere. And look at the mess this causes. This changes our whole reading frame. Instead of reading, let's see, 3. So instead of reading CTC and making this, and CTC and making this is like a tongue twister, we end up reading CCC. And then we end up reading TCT. And so every single amino acid down the line is wrong. And so we get a protein that's completely different from our original. And this mutation gives us a defective beta globin. The outcome is beta thalassemia, a genetic disorder in which a person has an abnormally low amount of hemoglobin. So our last one we have is E, an insertion of a nucleotide. So you look at this original strand, TCAGA, TCCAG. So we've added a base where it doesn't belong. And now our reading shift is we're going to cause a reading shift rather all the way down the line to the right from this point. And the protein is too short and it does not assemble correctly into hemoglobin molecules. And so you get, again, beta thalassemia. And so all of these changes we've seen have been bad. that's the thing. So that's not very good. So you can see that mutations are I'm not saying they can't be a good thing. I'm saying they're generally a bad thing. We'll talk more about sickle cell because that's actually kind of a cool example of some selective pressures with mosquitoes and malaria, and the heterozygotes for sickle cell are actually pretty good off. It's the sickle cell disease that are homozygous recessive, and that can be a trouble. But so maybe that mutation's not all bad. We'll talk about that in class. That's kind of I don't want to use the word fun. It's an interesting example. So here you have it, your last slide of the chapter. Why are ribosomes essential to protein synthesis? Not that tough of a question. Why is transcription necessary? Not that tough of a question. Why don't cells use their DNA as a direct model for protein synthesis? That's kind of a cool one. Yeah. Why do we have to make the RNA? Why can't we just send the DNA straight out? That's kind of a good question. I like that. And then in what ways does RNA differ from DNA? Very easy stuff. So I hope you learned something. Please take a minute and flip through your chapter 9 in your book. See the picture of the gross bald cat and see that sentence made of 3-letter words, and it is kind of cool. So take a look at that, and I hope you guys enjoyed it. So thanks for your time.